Good morning, everyone. And this morning we are going to talk about the plant disease diagnosis, but in a more practical point of view. And you will quickly see that making diagnosis in plant diseases, it's a little bit like making an investigation. So you will have to collect some clues, some information in order to determine which is the stress pathogen which is guilty for the abnormalities, the symptoms that you are observing. So you will need to collect a lot of information about the symptoms you have observed, but also about the context in which these symptoms have appeared. So you will have to observe the type of symptoms and their repetitions on the plant, but also the repetition of the disease plant in the field and the context of the apparition of the disease. So usually your observation will start with the plant. And you can observe the plant at 3 different levels, 3 different scales. So usually, firstly, you will observe the plant with your bare eyes, and you can then localize the symptoms and observe the type of symptoms that you have, the type of abnormalities that you have. And after that, maybe you want to go a little further, and you can use a more technical tool like a binocular, maybe to see if in the lesions you have some fructification, if you have a fungus, for example. And you can also go a little further again and take the fructification to observe them maybe under a microscope. So the first thing to do when you have a diseased plant is to observe the plant with your bare eyes and to see where are the symptoms localized on the plant. Do they localize on the leaves or on the stems of the plant, or maybe on the flowers or on the fruits? But they also can be on part of the plants that are not directly visible, that are hidden, like, for example, the collar or the roots or the vessels. So sometimes you have to dig into the soil to see the roots or cut into the stem to see the vessels. So sometimes it's a little bit more complicated. And sometimes it's also a little bit more complicated because the symptoms that you will directly observe will not be the primary symptoms, but secondary symptoms. So symptoms that are the consequence of a primary symptom. For example, here you have a cucumber plant which is wilting, and in fact the wilting is a secondary symptom which is caused by a primary symptom which can be damage on the roots or on the vessels, which are causing the wilting of the plant. So you have to be aware of that also when making your investigation. So you have located the symptoms on your plants. Now you will have to determine which type of symptoms do you have. So do you have chlorosis, necrosis, or other physiological abnormalities? And if you are really lucky, sometimes the symptoms that you are observing are really specific of a kind of disease. And in this situation, it's really easy to make a diagnostic. So here are some examples. For example, here you have the melon fusariose. So it's a fungus attacking the melon plant. And when the melon is attacked by this fungus, you will see a really clear pattern of chlorosis on the leaves, and which is really specific, really characteristic. So it's easy to make the diagnostic in this kind of pathogen. And it's also the same for some abiotic stresses, like the calcium deficiency of tomatoes, or, for which you will have a rotting of the tomato, which is also a characteristic, so it's easy to make the diagnostic in that case also. But in most cases, the symptoms are not specific. So you can have a chlorosis like this, which can be due to a large panel of stresses or diseases. So here on tobacco, we have chlorosis, and this chlorosis can be attributed to phytotoxicity, virus, nutrient deficiency, bacteria, and so on. So here, it's difficult just by observing the symptoms with your bare eyes to make a diagnostic. I've talked about, and Professor Daprich yesterday also has told about the biotic and abiotic stresses. But how can we make the difference between the two when you have plants showing abnormal symptoms? In fact, when you have a biotic stress, usually the symptoms are distributed irregularly on the plant because it's a living organism causing the symptoms. And you will also have a progression of the symptoms from a certain point, usually. And when it is a biotic stress, you will also have a species specificity. So if you have several plants in a spot, only one species will be attacked by the pathogen. And of course, if you see fructification in the case of fungi, you can see that it is a biotic stress. And when you have an abiotic stress, there you will have usually a regular distribution of the symptoms on the plant with no progression. The symptoms appear and that's all. And usually in that kind of stresses, all the plant species in an area can be touched. It's not species specific. For example, here you can see an abiotic stress and a biotic stress. Here you can see that you have necrosis which are really specifically located on the plants. Sometimes you will also observe that maybe the youngest leaf, only the youngest leaf of a plant is attacked when there is a deficiency or an excess in an element. And on the other side there you have a fungus which is causing Septoria api on cellar. And there you can clearly see the irregular repetition of the symptoms and the fructification in the middle. So you have observed the plants. Now you will observe the circumstance, the environment of your diseased plants. So it's really important to see which is the repetition of the symptoms in your field, in your greenhouse, in your aquaponics system. Is the plant you're observing the only one which is sick, or are there several? And looking at this distribution is really important because it will give you information about the nature of the disease and the way it spreads. For example, here you have a barley field in which you have really randomly dispersed yellow spots, and this is really characteristic of a disease which is spread by insects. Here, for example, it can be a virus spread by aphids. So it's really characteristic. Observing this kind of pattern can give you really important information on the disease you have. Here you have another example where you have a really straight and delimited band with yellowing plants, and just beside it you have plants that seem healthy. And you have, when you have some regular pattern like that, it usually has an entropic cause. So it can be a sensitive variety planted just beside a resistant one, or maybe it's a machine applying fertilizer or phytosanitary treatment that has had a dysfunctionment or something, and there was an excess of product on one band only. Last example here is a yellow spot which is growing from year to year in the direction of the soil, the working of the soil. And this can be the, we can have here a fungus, for example, or a pathogen which is spread in the soil. So it can be characteristic of that also. I don't know if you have some questions. Okay, so you have observed the repetition of the plants, the disease plant in your field. Another thing that you have to do now is to know things, have information about the context. So you have to go and ask the grower, or ask yourself if you are the grower, a lot of questions about the context in which the plants were cultivated. For example, is it the first time that symptoms like that appeared in your culture? And if not, what was the disease that caused the symptoms that you have observed earlier? Or what was cultivated earlier in that specific system? Because some parts of fungi can stay a really long time in dormancy in systems and can reattack new plants that you have planted if they are sensitive to the pathogen. So it's really important to know which plant was cultivated earlier in the system. The variety that you have used is also important to know, because some varieties are more resistant or sensitive to some kind of diseases. What was the origin of the seeds or the plants that you have used? Because some seeds or plants are certified free of viruses, diseases. So do you have used such kind of certified plant or not? Did you apply some phytosanitary treatment or fertilizers that could explain some abiotic stress or deficiency? Did you sow some insects in your culture that could explain the spreading of a virus? And so on. There are a lot of questions that you have to ask yourself to collect information to know more about the potential disease that you have. And now that you have made some observation, now that you have raised and answered some question, normally you should be able to make one or several hypotheses on the disease or abiotic stress that you have. With all the knowledge that you have or that you have in the books or on internet, you should be able to raise some hypotheses. So in most of the cases, the diagnostic can stop here, because you have a good, if you have a good hypothesis, you will not have to go further. But sometimes, if the disease is not at the good stage for making some interesting information, or if you have a viral disease or bacterial disease which are more difficult sometimes to diagnosticate, then you can choose to go a little further, but you will always there ask yourself about the time and the money that you want to spend in that diagnostic, but also what accuracy you want in your diagnostic. Do you want to know only if it is a biotic or abiotic stress? Do you only want to know if it's a bacteria or a fungus? Or do you want to know precisely the genus or the species, the complete species of the pathogen which is attacking your culture? So depending on that and the time and money that you have, you can eventually use the different techniques that we talked about yesterday, like an isolation and culture of the pathogen attacking your plants, to confirm that your pathogen that you isolated was really responsible of the symptoms you observed. You can make a Clerck postulate. And if you want really something more precise, you can use serological or molecular techniques in certain cases. But it really depends on your objective. Do you want to make a certification? Do you want to make a phytosanitary treatment? Do you want just to know it for science? So it's depending on your objective. Voilà. I don't know if you have some questions about all that. Was all clear? Okay, so what we propose now is to make a little roleplay game where Gilles will put you into a situation and you will have to ask yourself questions and have a reflection in order to make a diagnostic in lettuce.
To illustrate that Caroline has just said, let's do a little role game. So here you are a plant pathologist, and an aquaponic producer contacts you about a plant disease in his system. And your role in this game is to diagnose this disease. So for this, you need to make some decision, and this decision consume time and action. When I say action, is for example the cost or a move, etc. So the grower sends you this picture. Okay, you don't see something very precise. You see perhaps stunted growth here, smaller lettuce, but you don't see and you don't have a lot of information. So you need to contact the grower. So first you need to make a choice between simply call the grower or visit the system. If you call the grower, you can ask him some question, so by phone, and ask sample. So the grower can send you some sample in your laboratory, but it takes some time. You need to wait 1 day before we save the sample. And by visiting the system, you can make your own observation. So see by yourself the symptoms, the localization of the symptoms, and so on. And you can, for sure, ask questions to the grower and take sample for eventually some laboratory analysis. So now, what you think is the best to do? Call the grower or visit the system? Okay, the best is visit the system to see by yourself the fresh tissues. So you choose to visit the system, and I'm the grower, and you ask me some question about my culture. So I listen you. You want to see the system? Yeah. I answer of all the questions with the next dia. So, other question? What's happened? Yeah, since when? Sorry? Insect? Mm-hmm. The symptoms? Yes. What about the symptoms?
Well, for example, do we see fructification or like symptoms that would indicate whether it's maybe like a physical or a pest
Mm-hmm.
cause of pest or
Yeah.
physical.
So you ask to see the symptom on the plants.
So just physiological stress, like
Yeah.
temperature.
And perhaps the repetition of the symptom on the plant, the leaf or the wood, etc. Yeah.
Any changes in the water quality?
Water quality, yeah.
Control.
Control, yeah.
Are symptoms universally spread across the system, or it's just local?
Yeah, the repetition, yes.
Did you notice that before?
Mm-hmm.
Did you visit any other farm or institution or grow
Mm-hmm.
greenhouse where he might have taken the disease with him?
Yeah, for example. Also, perhaps about the lettuce variety. Mm-hmm. Okay, so let's answer to your question. Yeah. Okay. So for the answer, it's a one-loop WAF system with no complementation. The fishes are tilapia, and it's an important information. The grower has used a new lettuce variety this year. So this is a big information. The lettuce has 1 month, and most of the plants are affected in the systems. The repetition of the symptoms are not very specific, so yeah. And what about the symptoms? Sometimes leaves seem a little discolored and slightly wilt, but it's not very specific symptoms. Okay, there is a disinfection between each cycle, no UV disinfection, and another important information is what a power failure appeared 2 weeks ago. Sorry? Yeah. And this 2 past weeks, hot weather was also present. No insect in this case, to simplify the case. And here you have to see the roots, and you see that. So brown and necrotic roots. So you can say it's a rot roots. Okay? So you see that it's a big symptoms. And so the discolored leaf and the wilting leaf are a secondary symptom of this. Okay, and now you need to make some hypotheses. So if the symptoms are specifical of abiotic or biotic stress, and if it's a abiotic stress, it's caused by what? And if it's a biotic stress, which organisms or microorganisms are able to produce this kind of symptoms? And the last question is if you need to make a laboratory analysis. Usually we don't make a laboratory analysis, but if you need to certify your plant, you do some laboratory analysis. So now you have in your disposition some documents. So based on your knowledge and this document, you can look which pathogen or abiotic stress is responsible of these symptoms. And in the same time, you can already think about what kind of laboratory analysis you can ask or you can do, but if it's necessary or not. So you can do a binocular or microscope observation, and here you have, in each time you have the number of point of action and number of days to make the analysis. So you have binocular and microscope observation, analysis of water quality, lettuce composition, isolation and culture, and perhaps here you need an apiary or not. And serological techniques, so you need to know for which pathogen is it. The same for molecular techniques, and you can also do Koch's postulate. If you choose to make a laboratory analysis, what is the best solution? With less cost or the less time to do that? The best choice. Yeah. Yeah, if it's a fungus, we can easily do a binocular or microscope observation. It's not a lot of cost or time. Okay.
Isolation and culture.
Mm-hmm.
That's easy and relatively fast.
Mm-hmm. So yes, okay, we can check all the laboratory analysis. Yeah. So first, for the binocular observation of roots, you don't have really fructification, so it's difficult to see by binocular. The magnification is not enough. We can say this in this case. Analysis of water quality or lettuce composition, it doesn't make matter because the stress is biotics. So this analysis don't help us to identify the problem. If you choose to put in culture, first, if you need to know if you have an apiary or not. So for you, the answer is here, but sorry. So in what pathogens you think? It was Pythium? So yeah. So with a selective medium, so see you think it's a Pythium, you can do a selective medium. And if your petri dishes grow, you can say, yes, Pythium is present on my wood sample. But it's not sure that it's another plant pathogen. Pythium can be simply present in your woods. Okay? But if you do the same isolation, but without a priori, so you use a classical medium like PDA, you can have several pathogens. And you need to identify each pathogen. And after, you need to make a morphological identification of your culture. So for a microscope identification, you are going to make an observation of different morphological criteria. Like for Pythium, you will see sporangia, zoospore, and the oospore, and Enteridia, for example. Here you have a picture. So, but sometimes it's a little bit difficult to identify because you have some variation between different species, but also inside the species, in function of where you have isolated, or because some species or strain don't produce sexual spore, so oospore. So here, zoospore is the asexual spore of the pathogen, so they can swim in the water. And oospore is a sexual form of the spore for the conservation of the pathogen. And you, in any case, you don't have in each time this oospore, for example. So it could be sometimes difficult. And furthermore, some environmental or physiological condition can also make variate your final observation. So based on your observation, you can say, yes, it's probably a Pythium species. If you have choose to do oceological techniques, generally we don't do that for fungi. It's preferable to do a molecular analysis. But here you need an a priori to do this analysis. So you need to choose, yes, I do a PCR for a Pythium species. And here the results are positive. So you can say, yes, there is Pythium in my wood sample. But if you want to be sure that Pythium is responsible of the symptoms that you have observed, the best is to do a Koch's postulate. But you need a long time to do that. So you isolate the pathogen, you do a culture, you make your observation by microscope, and you identify the pathogen. After, you need to inoculate healthy woods and see if the symptoms newly appear. And after, you need to do a new isolation and one more time make an identification of your pathogen.